get going here. All right, everyone. I'd like to introduce Chi May. She's an associate professor at the University of Wisconsin, and she does a lot of development of biological systems, modeling, and does a lot of wet lab work. And she likes to investigate, the fundamental biological processes. And so without further ado, here she is.
Thank you. I appreciate it. And thanks for the opportunity to give this talk. And thanks to Herbert and everybody for for organizing the summer school. So I'll go ahead and start sharing. I think this is it. And okay. Let me go to presentation mode and here's my presentation. Oh, there it goes. All right. Getting confused. All right. Can you guys see the presentation?
Yes.
Okay, great. All right, so today I'm going to talk a little bit about some of the stuff that my lab does, and I appreciate everyone for joining in. I'm going to talk about some multiscale models that we developed in terms of the microbes and looking at infection and disease. And I'll try to walk through kind of step by step how we did some of that. And as I run out of time, I'll probably speak through the end or skip a section. But the real point here is to kind of show you guys how we're integrating, systems biology modeling at the molecular scale all the way up to the cellular scale, but specifically how we're applying it to ask some questions related to infectious disease and things like comorbidity. And so just as Bart said, at the center of what my lab does, we are a group that looks at trying to develop predictive models that ultimately enable understanding and kind of prescriptive design and programmability, whether it's microbial communities or host pathogen interactions that impact disease. At the core of me, I'm a computational biologist. And so kind of what my lab does is develop computational platforms, but these are very much informed by what we call measurement platforms. So methods that allow us to pull out temporal data or spatial data that we can then feed back into our computational platform, as well as empirical methods, experimental platforms and methods that allow us to either sample things at a higher temporal scale or ask questions that generate data that can further inform our computational model. So again, these are kind of iterative processes that we use to inform different things. And this is just a quick snapshot of different areas that we've worked in and the kind of work that we do. We really focus on multiscale models of host susceptibility and pathogen dynamics and disease. Heavily focused on TB right now, but we're also interested on the other side of the page on Francisella tularensis. These are both pathogens that cause structural immune response. So there's a granuloma component to them. So that spatial immune response is also important in addition to that chemical response as well. And so some of the work that I'll highlight today is work that was done by Cheryl Sershin, who's a former postdoc in my lab. And so some of the experimental work has been informed by Maya Goff and Komal Rasaputra, also former members of my lab. And then on the right-hand side, there's some spatial work that I won't get a chance to talk about, but I'll get a chance to talk a little bit about Taha Salim's work in developing some models of macrophage response. And so some of you might be quite familiar with tuberculosis or the process of infection. So Mycobacterium tuberculosis is one of these pathogens that highly infectious. It's airborne, and it can, once it infects someone, of course, then you can pass it to somebody else. It resides in the lungs. It's a very oxygen loving pathogen, and one of the primary immune cells that takes up phagocytosis Mycobacterium is called the macrophage. So this macrophage is a critical immune cell that not only is involved in innate immunity, but helps kind of bridge that gap between innate and adaptive immunity, kind of the two arms of immune response. So in terms of importance, it's a top 10 cause of death worldwide. So it affects a number of people. There continues to be new incidences, oftentimes in developing countries, but it can also be a concern within the US, and specifically when you have comorbidities that then predispose you to that. And the other component of concern with TB is the rise, like in many infectious diseases, of resistant to some of our first line drugs. So multi-drug resistant TB continues to be a concern. And also something that I won't talk about today, there are disparities in terms of populations that seem to have a higher prevalence of TB, even in the US, and there could be comorbidities that are associated with that. One of the things that we are very interested in looking at is vitamin D and alcohol use, and how that deficiency coupled with alcohol use actually impacts that, both modeling it experimentally and computationally. So kind of the premise that when we're trying to consider things, we're trying to consider how the rate of response of the pathogen, the rate of adaptation of the pathogen, which I'm kind of showing on that y-axis, compares to the rate of response or immune response of the host, which I'm showing on the x-axis. So how quickly is the host creating a negative environment for the pathogen, and how quickly can the pathogen adapt to this negative environment? And so we're, kind of the underlying hypothesis that we and perhaps others have been considering is that, okay, that rate might be important in terms of when we have an unaffected granuloma or construct that then allows the pathogen to grow and continue to be active, versus when we have kind of an effective granuloma that protects the host and can also be protective for the pathogen, kind of this granuloma formation that results in latent disease. Or if let's say the host is very robust and it's very effective in creating a negative environment that gets rid of the pathogen, it's what we call clearance. And so kind of we want to relate this robustness of the cell immune response to the pathogen. And so with that, we're developing models around the microbe. We're developing models around the bacteria, its metabolic response around the host, the biochemical response of the host, and then the environment that the host is creating, whether it's that microenvironment inside of the host tissue or elsewhere. So we're trying to kind of integrate all those. And so kind of, I know you guys have learned how to model systems at different levels. So we're kind of trying to integrate I'll show you some stuff where we're looking at gene network models to more of, when I think of cellular networks, I'm like signal transduction pathways, metabolic pathways, and ultimately kind of where we end is kind of our cell-to-cell interactions. And so I'll start quickly and discuss models of the microbes. And I'll try to keep an eye on my time here. So the first I wanted to start with is just kind of talk about TB. And this is really where I entered in and kind of where our model starts. And we're really interested in TB's metabolic adaptation to oxygen. And the reason being, again, as I mentioned, TB is this lung pathogen. And so oxygen is one of the one important factor. And so the first model that we developed was based on the experimental work by Wayne and colleagues. So there's this Wayne model of non-replicating persistence, because one thing that TB does, I talked about latency. So TB kind of goes to sleep, or I don't know if it's necessarily going to sleep, it's still metabolically active, but TB tends to, it persists over time for a long time. So you can be exposed, not even know it. And later on, let's say you get older or you get some other severe like co-infection that it compromises your immune system, it can reactivate. And so that's been this characteristic of TB. And one of the models of that is this Wayne model, where they expose TB to different levels of oxygen, either well oxygenated, which I'm kind of showing with this test tube here, or low oxygen to no oxygen. And what the Wayne model found is that TB can survive as long as the rate of oxygen depletion is slow. But if that rate is fast, TB fails to survive. And so the question is metabolic, or why? What are the biochemical reasons for that? Of course, they found certain things such as the activation of glyoxylate, the increased production of glyoxylate. And so one of the things that they found is that this glyoxylate production could be shunted through the glycine dehydrogenase pathway to produce more NAD plus. And so we wanted to kind of ask the question, how does this impact the production of ATP, which was something that was measured in the study. And what the Wayne study had found is that ATP kind of holds its place when TB survives, but it drops when it does not survive. And so we connected the TCA cycle metabolic model with this glyoxylate to glycine shunt model with the electron transport chain. And the electron transport chain takes some of these cofactors, and in the presence of oxygen, it's able to take it through the electron transport chain to produce ATP. And so that was our way of connecting the different pieces that were in the experimental studies with the different biochemical and metabolic pieces that could be underlying the reason why this works or doesn't work for TB. And so just to quickly say that, and so as many modelers do, we collect pieces of data from different spots. And so one of the key pieces of data that we wanted to integrate with the Wayne data that showed us things like ATP measured growth under well aerated conditions, under different conditions where you have slow oxygen depletion and rapid oxygen depletion. We integrated that data with data by Voskuil and colleagues where they actually measure the gene expression over time. Now, this type of data is not only found in the papers and like supplementary materials, but also like in geo databases that are available. So we pull this data and we combine the data with, again, measurements such as growth, such as ATP levels that were reported in the paper. So we use things like Rabbit to pull the data and things like that. And we put it together in a regression-based model, both linear and non-regression based, because our question was, how does the oxygen level impact gene expression, now impact metabolic response, right? And so this is just an example of just how we pulled some of the data. And there's some, as you're doing this, if you're not generating all the perfect data yourself, there's some correlation where you have to say, okay, this is how I normalize between papers and between data sets. So we did some of that. And then I just wanted to show you guys kind of what it looks like. So we basically, out of this aggregate data, we have things like time. We have fold change of our gene that we're trying to model. We have change in time, change in the environment that we're trying to drive the gene model with. And so we pull all of that together and then we come up with regression based, these are statistical models of gene expression so that we can now say microenvironment to gene. And then this model of, let's say, gene ACEA goes to inform the enzyme model of that then catalyzes that metabolic response that we're trying to model here. So everything in red, and I know it's hard to see, but everything in red, those are enzymes. And so these are the genes that correspond to those enzymes. All right, so then we put together the model. And so this is kind of an example of the enzymes that were modeled and the reactions that are modeled in there. And so we have, some common reaction forms, which you guys might have already seen during the course. And so most of their, since these are metabolic processes, we're modeling using Michaelis-Menten reactions. Some of them are reversible, some of them are not. And so sometimes you have single substrate, multiple substrates, multiple products. But so we have common forms of the rate equations. And then we tie it to our gene model using kind of this fractional occupancy approach that actually we pulled from Dr. Sorrow's papers in his book. And so what that allowed us to do was say, okay, if we can look at the ratio of all active forms of the genes that inform the production of that enzyme, and divided by all possible forms of the gene, we just basically get this number between 0 to 1, kind of this probability of the enzyme being active. And we use that to get this effective Vmax value that modulates the maximum velocity, because clearly the system isn't always operating at maximal velocity, but the fact that we can modulate it based on the incoming environmental dynamics actually gives us a better replication of our metabolic response to try to mimic probably what's occurring inside in vitro and in vivo. Okay, and so we did all that, and so this is just an example. After we implemented in something called BioZeis, which was a tool that I co-developed while I was at Sandia National Labs, and it's a circuit simulator, but we use the approximation of an electronic circuit simulator. The cool thing about that is massively parallel, which lets us do the next step that I'm going to talk about. And so, we used BioZeis to implement it. We optimized it using Dakota. And then we asked a question, how well does it match kind of some of the empirical data that we pulled in? It's kind of hard to see. We use a t-test to say how different are these data sets. And what we found is that in terms of, we were able to replicate the well aerated condition and the slowly depleted condition. And part of the reason for that is because the data was more available. But for the rapidly depleted condition, we didn't have gene expression data for that. And so that wasn't as well replicated, even though when we looked at the means of our values, they were comparable, but there was some statistical significant difference between them. But part of our question was, was to try to replicate that, but we knew that at least we were able to get some of the general means of the different data sets that we had to be more comparable. So on the right-hand side, I'm just showing an example of one of the substrates that we modeled and kind of some of the behaviors that we saw. One of the behaviors that we kind of observed was that it looked like when oxygen was rapidly depleted, it didn't seem like TB was, at least the network was not adapting quickly enough. Like in this example, I think the dotted line is rapidly depleted and the blue line is well oxygenated. You see that the rapidly depleted continues to mirror the well oxygenated conditions versus the red line is the slowly depleted. It seems to adapt differently. And so we kind of noticed that among some of the substrates that were replicated. And so we performed an in silico study to ask the question, what is this adaptation response and how does that impact the system? And so what you see what you see in this is ATP level. So when we look at ATP levels, and ATP synthase, which is that kind of final enzyme that then catalyzes the production of ATP. And I'm showing you the inset of the earlier time periods. We used our data and we divided our data into the same regions that the experimental model, the Wayne model, had looked at, which is active growth, NRP1, NRP2, and then fully kind of this anaerobic response period. And so those correspond to when TB has plenty of oxygen, but then you're going through this depletion phase and then anaerobic fully, there's no oxygen in the system. And so what we see in the inset here is that the blue is the when slowly depleted oxygen conditions, the red is when it's rapidly depleted. And so you see this difference in ATP synthase production, right? So the slowly stirred where it survives, its survival curve, it's increasing ATP synthase much earlier. But then you see at the very end where it's actually dying this rapid depletion state, it actually increases ATP synthase, but it's way too late. And so kind of what we found is that there is this reprogramming of the TCA cycle to enable TB to kind of adapt. You see under this, when it actually survives, it has this positive adaptation. So this is where the red, so you see things being activated a little bit earlier. And these are enzymes, right, that are part of that metabolic pathway that we were looking at. Versus you have this in the rapid system, it has this negative adaptation response. You see 7 enzymes during active growth that are kind of downregulated. And so you also have this retrofitting to regenerate more NAD, which then can be piped to ATP production, which we see under the slowly stirred condition. So the importance of this study was kind of twofold. One was that no one really had data on the rapid response. And so we were able to, based on existing data, build models that we can then now study and more look at the molecular response of the rapid under that specific condition. And we saw this clear difference in what it was doing. Now, of course, need to go. Somebody, maybe someone's already done the study and we can look to see where we write, or maybe we could do the study, but we haven't yet. But it just kind of shows the power of kind of pulling together existing data to then say, okay, what can we ask with our empirical studies, with our experimental, our theoretical studies, right? How do we use these simulation-based approach to do theoretical studies? And then the other thing, this was part of a paper that Herbert was one of the co-editors where they had a special issue on reproducibility. And so we actually went through the exercise of taking this model, putting it as BML, and it was reproduced by a student or someone in Herbert's group. And so that also was, for us at the time, it was something that we had not done before, and it kind of made it more readily available and showed that the model could be reproduced, that we had provided enough information to do that. So these types of models, I'm sorry.
Sorry to interrupt, Gigi. One of the attendees had a question in the chat about how you select the enzymes of interest in the heat map.
So the enzymes that this might be all I think these are all the enzymes that are in the model.
Oh, period. Okay.
Yeah. So I think those are all the enzymes in the model, I believe, or they might just be the ones that are in the ETC channel. But I think they're all the enzymes in the model that we looked at. Now, these are made up of a whole bunch of genes, right? So all those gene models come together to form, because you need multiple genes to form the enzyme. But this represents the actual enzyme. Yes. Is there another question?
Yeah, Stephen? someone has their hand raised. Oh, yeah. Nope. Oh, how is Y calculated in the model? Which Y
So,
is,
yeah, so I think it's let me go back. So this is the Y. So this is
oh,
active. Right.
yeah.
So this has to do with, so each of these like ACEA, there's ACEA and A sub B. Those are all components of, you see these red, these enzymes, right? Right. So these enzymes are made of multiple genes. And so it's a ratio of all active forms of the gene divided by all possible forms, right? The ones, so in, maybe I should have gone back and said this. So we pulled, there's a reconstructed metabolic network of TB that was developed. So this is used a lot of times to do like FBA types of modeling. And within that, there are gene relationships that say you have enzyme A, Boolean relationships between gene components that are supposed to be used to produce the enzyme or that specific protein. And so we pulled those relationships. And those relationships are also available in like biopsych databases. So using that, we formulated a Boolean representation of the enzyme. And so active forms are based on what is in that Boolean representation. So what they say is you need like gene 1 and gene 2 for the active form. But let's say it's you might need gene 1 and gene 2 plus gene 3. Right. So that would go in the numerator, and then the denominator would be all possible forms of it. Right. So this is a ratio. Okay.
Yep.
Great questions. Really great questions. Um, So Bart, you're gonna have to cut me off. I know, because there's no way I'm getting through this in 15 minutes. So I'm just gonna get through
Well, that's
the
all right. Just, yeah, focus on, try to focus on the important parts.
all right. I'm gonna talk about the last piece I'll talk about will be how we're integrating this. So I just wanted to briefly mention that these types of models are, clearly many different things, including we've looked at something called systems chemical biology, where therapeutics, right? If you have this model of the pathogen, you can then now say if a chemotherapeutic small molecule targets something, what happens to the system? And we've done that, and kind of, does it impact ultimately ATP production? Is it a good target? So there's translational impacts. And then also just to say that we're using this approach to modeling to look at different things such as microbial community. This is something that one of my students, Majid, put together, and Daniel Ajouzi is continuing to work on it. Had a postdoc that worked on it, but this is really looking at microbial communities, and each of those little dots, they have a metabolic model that's also gene driven. And so you can, appreciate this, and we put it together in a 3D multi-scale model.
Mhm.
And so this can get computationally intensive, but it's important that we can now hopefully use it to ask interesting questions. Okay, so here's where we wanted to go with this. We wanted to add physiology to some existing multi-scale models of TB. So as I mentioned, TB forms this granuloma, and Denise Kirshner's group has been for a long time building multi-scale models of TB, multicellular and multi-scale models of TB. And so there was a 2009 version of their model it was a great model in terms of specifying all these parameters to use an agent-based model to try to look at granuloma formation in TB. And so starting from that model, we wanted to add a couple of things that were not in that 2009 model at the time. One, we wanted to explicitly add in oxygen. So because this is, in the lung environment, we wanted to understand the impact of oxygen on the cells that were being modeled in that initial Kirshner model. And then explicitly model TB, because in the original model, I believe the way that the pathogen was modeled more of a field versus an explicit model of TB. So given that we have this TCA model, we have this connection to oxygen, Cheryl started putting together a 2D model of an agent-based model of TB response. So we called it ABM Phys, right? Just to highlight the physiological response part. And so what this involved were different things, again, going into the literature to find kind of these physiological properties, pulmonary blood volume, what happens when you breathe, right? How does that impact how much oxygen comes in? And so the way that ABM models work, and I believe later on you guys will hear more details about that. so you have there's some stochastic modeling to it. You have upper and lower limits, you have a distribution. And so Cheryl went through and based on the literature, based on trying to develop the physics of how oxygen comes in and how we should do how the diffusion model should be developed. There were certain parameters that were now added into the original base ABM model that was developed by the by the Kirshner group. So we actually re-implemented the Kirshner model as a base, and then we added our model. And so the tools, and so then we added TB, and of course TB specific growth rate and how that was modeled. And then the host, right? We had to also add host parameters that were associated. So the host as an agent and how that was associated with how these models actually responded to oxygen. One of the things that we explicitly did, so in the agent-based models, you have grids that represent space and tissue, right? We actually explicitly, for each of those grids, we represented extracellular bacteria with one of our systems biology models of the TB, the metabolic model I just showed you. And we explicitly represented intracellular bacterial load with explicit models. So each grid could have up to 2 different TB models ongoing to represent TB taken up by the macrophage or TB in the extracellular space. And then, of course, there were the rules that were associated with that. And so this is just to kind of show the flow, right? So you had first the execution of the rules of movement, the cells moving around the different grid points. And within those, we had different types of cells, including the macrophages, the T cells, and whatnot. Then we had the fields and the diffusion of some of the cytokines and chemokines that already existed, but then we explicitly had to account for diffusion of oxygen. And in lieu of having a field for the bacteria, we just had specific bacterial growth that we had to calculate based on our metabolic model. And so then after that, you had an update of the oxygen field. And then depending on those environments, we then ran the intracellular model for bacterial fitness to determine whether it was growing or not. And so this was run with BioZeis. So this is where BioZeis's ability to be massively parallel became computationally important. And so with BioZeis, we gave it inputs and we gave it outputs. Its inputs were like the substrate level that it previously had, gene expression. And again, that gene expression is now being driven by that oxygen field, right? And then the outputs were the ATP levels and NAD ratios that were then used to modulate growth rate. So we did it, that was tied together, and we ran this, did optimization, all those things, sensitivity analysis, and found certain things that were critical, such as threshold, pulmonary blood volume, all these things. But anyway, just to kind of show you what that looks like. And so this is an example of the simulation. You'll probably recognize this common simulation that you typically see in images that you see. So different types of cells going on here. This is an example of containment. So this is a formation of a granuloma in the center here. See some of the brown, which are some of these necrotic regions, and yellow. And this is the first graph. Yellow in that first graph represents some of the bacteria that could be extracellular, intracellular. Some of the key things that were added here. So this is our oxygen field, and I'll run that again. So this oxygen field just kind of shows where the oxygen is clearly. And then one of the cool things I thought is we can now look at certain things such as the rate of oxygen, because that was one of the things that in the Wayne experimental model they found was important. Then on the bottom here, what you're seeing is the delta extracellular bacteria. So this is the change in ATP in extracellular bacteria. So under containment, you see that it's kind of in the lower end, which is lower levels of change in extracellular bacteria. So there is extracellular bacteria, but the rate at which is, the ATP levels are kind of low. And you can contrast that by this one on the right, which is intracellular bacteria. So the blue means higher levels of ATP. But the cool thing for us is that, and for all of us that do modeling, I think kind of what we're trying to get to is we can actually go down, look at different things, observe different things using our models if we can put them together in a way that's robust. On the right-hand side, bottom right, what you're seeing here is then we kind of compared, this is total CFUs in our simulation. And the cool thing is that we did not, we didn't program this into the model. It is an emergent property of the model, because you see the CFU going up initially, the blue line, and then coming back down. So you see this containment profile which was the same trajectory that was observed in empirical experimental studies of in vivo infection using, I think, non-human primates and things like rabbits. The red line is the initial ABM that it was based on. This is the initial Kirshner ABM. I mean, by the time this came out, they already had better models, but this was the base model that we compared it to. And so just to show that this dynamics, again, it was not programmed in, but I think because we were able to explicitly incorporate that growth rate and treat each bacteria individually, I think that's part of the reason why this came out. But there's many different things you can get out of this. So just to kind of, we did the validation in comparison to this was a study out of the Flynn lab. So if you look at our rates, which is the thing in the pink reddish, of clearance occurring, containment occurring, dissemination occurring, it was comparable to what was observed in experimental studies. And on the right, kind of the level of the CFUs, ours is in the middle, was comparable compared to our base model the Kirshner model, the base Kirshner model that we used. And I always have to be I want to overly explain that they have great models. But this was the one that so basically we were able to replicate this without it being statistically significantly different, again, using those steps. So just out of this, so this is an example of when you have a response that kind of goes out of control. And as you can see, it doesn't look excessive, right? But we're able to kind of capture something. The yellow are extracellular bacteria. And so you clearly see here that on the right-hand side that the bacteria is much more active. ATP levels are much more higher. So the cool thing is that you can capture the dynamics of this immune response, right? And how oxygen, this physiological component, is modulating this. And we can link it to, so we're able to now link to structural immune response. We're linking our metabolic models, our gene models, to kind of the structural multicellular response. And one of the cool things that came out of this study is that we observed something that was a new outcome, right? So we thought that in the way it works, so Cheryl would simulate many different things, right? Expand the parameter space. And then post-processing is to go back and observe the outcomes and ask the question, does this bin result in containment? Does it form a granuloma for the time period that we simulate for? Does it clear the infection? Or does it, bust open, right? Does the infection disseminate, right? So those were the three bins that we were considering. But one thing that was observed, that Cheryl observed, was this new outcome, transient containment, which is kind of latency and reactivation, you can say, where initially she thought this was a containment outcome, but then as it continued to simulate, the cells, the bacteria started growing again and kind of reactivated. So this was this fourth outcome that was in no way programmed in. And so that was one of the cool things that came out of this one of our models. Okay. So I'll pause if there's any questions before I move on. I saw it.
Yeah, there's a chat. So the Y value has no relation to the expression level. For example, I need 2 genes expressed to make an enzyme, and there can be 3 forms. Then Y is 1/3. Wow.
No, it has an expression. It's not just simply 1/3. It's the values that, if you recall from the regression model. So the regression model is an equation. And so that equation has a value, right? It's not just a simple integer. And so you take that equation, right, those real numbers, and then you look at the ratio of those real numbers. Yes, we normalize it. So ultimately you have a number between 0 and 1, right? Because you can't have higher than B max. But it is based on that initial regression model of the gene.
Okay, there's another question. How are the metabolic cell growth parameters linked in the model? What about the cellular communications between cells?
I'm assuming this is the cellular growth. So this must be just the model I just talked about, the multiscale model. So the way that the cells, so within one agent, let's say the macrophage, right? So as it can produce, there's a rule for it to produce different cytokines. And those cytokines go into the field model for that cytokine, let's say interferon gamma, right? So the macrophage can produce interferon gamma at a certain rate, depending on whether it's infected or not. That production goes into its grid cell. So now its grid cell has a certain concentration of interferon gamma, right? And so then we have a field model that calculates, what's that concentration, what's the probability of that one of diffusion to another grid cell, which then impacts another macrophage. So it's through that what I call that environment model. So the extracellular environment model. That's how they're kind of communicating. If I hope. Yep. Okay.
And there's another question here.
Okay.
It says, I think you said that the slow rate of oxygen depletion was the time for gene expression to readjust. Is this the slowest time scale? And how long does it take?
So the slow rate, so oxygen depletion rates, so this is again that first model. So those oxygen depletion rates are based on experimentally determined levels of oxygen that the Wayne experimental model had measured, right? And so that rate, I don't remember what that exact rate is, but yeah, so basically what we're feeding in is different levels of oxygen so when we're actually running the model, we're saying, okay, this is your oxygen, this is how much oxygen it's in there. That first model, we use the Wayne values to say, this is how much oxygen is in there. Now simulate and give me your response. But in the agent-based model, we don't know what that rate is. It all depends on what's going on with the agents, how quickly the macrophage is coming in there, how quickly it's forming these environments. So that's not predefined. But we used the predefined Wayne model to first build our metabolic model. Hopefully that clarifies it a little bit more. Okay. There's one more.
Yeah, I think Aditya. Yeah.
The question which I was asking was like you are modeling metabolism as well as you're modeling multiple cell types. So these are two independent models, one after each step they are getting integrated, or like they're running parallelly and they are getting the model as a single entity in
Okay,
the
good.
previous one.
Yeah. So this is the kind of the flow. So the cells themselves, the cell model responds, right? So once we set you have an initialization. So the cell models, they move, they respond, right? They determine whether you start with an infected cell in the middle. So the cell response is kind of that rule-based, more, I would say, coarse-grained model. So they respond, they do certain things. Then we set based on where the cells are, we're going to calculate our fields and differences in the field, and we're going to calculate our oxygen field. We're not running everything in parallel. We're going through a loop. So there's kind of a master that calls each submodel. And then once the cells are kind of set, then we calculate the fields and the oxygen field, right? So now all those conditions are set, and then we calculate the explicit ODE model for the bacteria.
So these are at different time scales.
So there's a kind of a master timescale. So they're different. Well, they're okay. So there's the computational implementation of them that they're running. They're not running concurrently. They're not running in parallel because you have to communicate between them. Then there's the actual timescale, the simulated timescale. They're running within the same simulated timescale. So like if we run, I don't remember the exact number, we might run for 10 minutes, right? This movement represents 10 minutes of time. We then run the ODE simulation for 10 minutes. And then we report back the value.
Okay, got it.
All right. Okay, so the last part, if we don't have any more questions, Bart, can I continue?
Yep. Yes, you can.
Great questions. It's great that it's interactive. Okay, so the last piece I'm gonna, I think, be able to chat about is models of disease. And so what I'm calling it, of course, infection is in of itself a disease, but other comorbidities that now impact immune response. And so because most of us, we're not just walking around a picture of health, right? There's different things that impact your susceptibility to infection. And we, heard about this during COVID, right? Age, pre-existing conditions, things like diabetes, HIV, and things that we might not think of, such as as I mentioned, very much like looking at vitamin D deficiency. How does alcohol use coupled to that? One thing that we started looking at is things that are more related to oxygen, such as chronic obstructive pulmonary disease, which is COPD. And so Cheryl built a model around this to say, okay, how does that comorbidity now impact our TB response? So just a few things in the US, there's, COPD is an issue. And one of the things, there is some disparity in terms of who struggles with COPD. Lots of different people do, but one thing to point out, Native Americans have a slightly higher level percentage of COPD in their population. The other thing that also can cause further damage related to COPD is smoking. And sometimes smoking rates can also vary, especially based on income. So there's this intersection of not only comorbidities, but health disparities among various populations and their risk for, the fact that they have a comorbidity and how that impacts then their outcome, and things that we probably should start considering, or people are considering in the healthcare industry, but we think that we can help develop mathematical models around that. And so looking specifically at COPD and smoking, we took our ABM Phys model and added certain things to it. So one of the things that Cheryl found in the literature is that COPDs, they have triple the risk of developing active TB. And COPD itself impacts certain things such as how macrophages and T cells are recruited, pulmonary dynamics. So because you have damaged tissue, your oxygen distribution and diffusion is not going to be exactly the same. Your cytokine, the inflammatory environment within the lungs, the cytokine chemokine levels, and metalloproteinases, and whether they're present or not, and how that can cause further damage. And so certain smoking also can, again, contribute to that. And so she, starting from that original model, collected additional parameter data based on experimental data, and well, I guess a lot of empirical studies on, or maybe their epidemiological studies on different patients. And so we added into the model, she added certain things, including a goal stage, which has to do with kind of severity of COPD and how that impacts age, the oxygen, and different things, MMP8 and MMP9, and I think MMP9 was the main one that she added, but these MMPs we didn't have previously, which had to do with the metalloproteinases that were in the environment, because then that impacted tissue damage. And so one of the ways that, and then she ran the simulations. And so what you're seeing, just to point out quickly, she looked at COPD. So all of these patients have COPD, but this distinction is how much damage they have based on COPD, whether it's low damage versus high damage COPD, and then whether they're a non-smoker, which the top is. So the top left, non-smoker, low damage; top right, non-smoker, high damage; bottom, light smoker, again, still with COPD, and the other one's a heavy smoker. And so what you can see, this is just a representative granuloma at 200 days. So you can see that the non-smoker, low damage, that the forms of the granuloma are slightly different. And clearly this heavy smoker with the damage, the black is the areas where you basically have dead tissue. So you're not going to have cells there. You're not going to have, as much of the oxygen isn't going to be there. And so you kind of see we do have these different formulations of COPD once she reruns when the studies were run. The simulation studies. And so one of the other things that you can go in and do with some of these types of models is now ask questions about the cell populations. And so when we look at the cell population, this is the average number of infected macrophages. For instance, this is just pulling out one of those sample data points. What we saw that was interesting, when we compare the average number of infected macrophages to the average number of cytotoxic T cells, and cytotoxic T cells tend to be the ones that, like the CD8 cells, that signal for cells to die. We saw that macrophage response aggregates differently than cytotoxic T cell response over time. So heavy smokers and non-smokers with heavy damage tend to aggregate together compared to light smokers and non-smokers with low damage. So the heavy smokers and the non-smokers with heavy damage are the ones in the black and the red. So you see an increase in the average number of infected macrophages. Versus the T cells is kind of the opposite. They aggregate based on smoking. So heavy smokers and light smokers, which are I think the blue and the black, they kind of have lower numbers versus the non-smokers and non-smokers with low damage. I hope I said that. Yep. So they have higher damage. They have higher numbers of cytotoxic T cells. Don't know why. We haven't dug into it really deeply, but it was just an interesting thing in terms of how these different cell populations aggregated differently depending. So which suggests some type of, functional aspects to this disease. But when so looking at in terms of after the simulations were run, she compared COPD to non-COPD cases in terms of outcome. And so typically your outcome in orange is a non-COPD. You would expect most of your individuals to clear infection. And then another portion, smaller portion to end in containment, and very few to end in dissemination. But you see kind of a difference in COPD. You actually have very low clearance, and you have most of them resulting in containment, which would be granuloma formation, and then even more resulting in dissemination. And this was more comparable to what's been reported in what was reported in the literature, which shows you that this comorbidity is extremely important to consider as you're thinking about TB treatment and things like that. And so that's going to be where I stop.
Okay.
And I just let me just put up my thank you slides and that's it, and just acknowledge those people.
There's a couple questions
Okay.
on
right.
what you just talked about. Are the simulations validated using experimental evidence? Are they using flow cytometry or spatial transcriptomics with cell profiling?
So for the COPD, no. So in terms of, we haven't looked into the literature to see what they say in terms of the number of, let's say, CD8 activated CD8 cells. That's a good idea. But we did validate them against like the numbers of reported, kind of that last bar graph I showed you, kind of the outcomes. So compared to the outcomes, you do see higher containment in COPD patients. So that was how we validated that.
Okay. And then there was one question going back to the first model you're talking about, and a continuation of how did you pick the 10 minutes as time for one iteration on the previous time scale discussion?
Yeah. I don't recall how exactly we picked it. 10 minutes. I don't remember why we chose 10 minutes, but I know I don't recall whether it was a computational constraint or just based on how quickly, in terms of diffusion, it might have been related to oxygen diffusion, actually. How quickly we could measure oxygen diffusion and how quickly we simulate it. That's what I'm if I recall. That's what I'm thinking. Yeah, because one of the limiting factors is diffusion, right? Things diffuse so quickly. We want to capture our system in a place where we see the dynamics of diffusion, right? And have that dynamics actually impact the cell dynamics and propagate down. So we sometimes we are limited by how quickly things diffuse.
Okay. All right. Well, thank you very much, Gigi. We appreciate you spending some time with us.
Oh, I appreciate it. Thanks for the opportunity and the great questions.
All right.